The topic for the panel will be the putting the patient first and last, using data and tools to accelerate therapies at the clinic. We've already introduced Bonnie, and Bob, when he comes, he's been introduced as well. So let me say a few words quickly about the other panelists. First, we have John Raffa. There's Bob now. Sitting next to Bonnie, we have John Raffa, who currently serves as the Executive Director for Research Services at the Avoca Group. He'll be speaking today, and that is a company that provides comprehensive relationship management, consulting, and research services to organizations in the pharmaceutical and related products industries. Prior to joining Avoca, John was in JR3 Consulting, delivering analytical consulting services to companies in the life science industry, and spent many years at Covance providing competitive intelligence, market research, and strategic decision support to stakeholders at various levels. John's experience also includes several years of consulting. He has a BS in marketing from Villanova and an MBA from Temple. Next is Michael Cantor, Dr. Michael Cantor. He is a Director of Clinical Research Informatics and Associate Professor in Clinical of Medicine and Population Health at New York University School of Medicine. As Director of Clinical Research Informatics, his work focuses on optimizing clinical data for use in research and quality improvement. He's also a practicing internist. Prior to joining NYU, he was a Senior Director, Information Strategy and Analytics at Pfizer, and his work focused on the secondary use of clinical data, personalized medicine, and automated methods of pharmacovigilance. For Pfizer, Dr. Cantor was the Chief Medical Information Officer for the South Manhattan Healthcare Network of New York City Health and Hospitals Corporation based in Bellevue Hospital. Dr. Cantor has completed his residency in internal medicine and informatics training at Columbia, as an MD from Emory and an AB from Princeton. And the final panelist is Mariano Alvarez, an associate research scientist at Columbia University. He obtained his PhD in Buenos Aires, Argentina, studying the interaction of melanoma and host tissues. He then spent 3 years using functional genomics to study the interaction between the brain and tumors growing in the periphery in small biotech startup companies. He joined the Califano lab in 2006, where he gradually became involved in the execution and coordination of several projects related to the elucidation of drivers of pathophysiology. He used reverse engineering techniques to assemble network models of cell regulatory processes and develop methods to infer from such models the protein driving specific phenotypic changes from stem cells maintenance and differentiation to the dissection of the process driving neurodegenerative disorders to cancer initiation and progression and the inference of drug sensitivity response to multiple drug interaction. So with that, I'm going to turn the floor over to Bonnie, who will moderate the chair, will moderate the panel as chair, and I'll...
Okay. Do we have slides? Okay, so we're just going to have a few slides. And you've just heard about Judy Jetson and how we've got our innovative companies, SciAps, Numeti, Personalis, capturing the explosion of omics, and IASD and GNS Healthcare and DNA Nexus using new tools in big data research and development with Transparency Life Science, Zephyr, and Gobalto, and the Scripps Translational Research Institute working to speed up clinical trials. So if you think about the person, the panel is all about personalization and the fact that we're all different. I'm excited to bring us these 4 experts who are going to look at this problem from a variety of different perspectives. And first we're going to have John, who's going to give us she's going to look at the big picture and explain to us how we need to shorten research and development cycles in order to get therapies to the patient faster. Michael's going to tackle the problem with a different perspective and look at 3 areas. First, he's going to look at data, then he's going to look at tools, and then he's going to think about the patient. For data, he's going to answer our question of how do you make data sources analyzable, and tools, how do we give some examples of clinical decision support, and with the patient, how can we tailor our interventions so that it's all about me? I don't know how you feel, but when I go to the doctor, I want it to be all about me. Mariana's going to give us a research perspective and help us to bring this together. And then Robert, as his external role at J&J, is going to tell us how they think about the patient front and center, with collaboration as a focus of all of our thinking. So with this in mind, I want to encourage all of us to look around at your neighbor, get to know each other, see how we can collaborate, and I'm going to share a secret, a personal secret about each of the panelists, so that maybe when they get up here, you think you already know them. Let's see, John. So John tells me that he likes to think of himself as a strategic thinker, but recently, when he's been struggling to play chess with his 5-year-old, he's really wondering if that's the right approach. Michael spent the last semester of medical school in Paris at the Institut de Pasteur doing infectious disease research. And it seems like all the panelists have a theme about Paris. I myself love Paris, and it seems like they do too. Mariana's secret, he told me this morning, is hiking. So do we have any hiking enthusiasts? Me too. Okay. And then Robert says he loves all the same things I do plus 1. So the ones he loves are travel, meeting new people, mine is anything chocolate, and his is jazz. So, since we all love to visit in Paris, maybe we can meet up there. But for right now, I'll turn it over to John.
Thank you, Bonnie. Just for the record, I can still beat my 5-year-old at chess at the moment. He's gotten pretty good, though. So, putting the patient first and last, using data and tools to accelerate therapies to the clinic. That's the focus of the session today. The focus is accelerating therapies to patients who need them. So let's start just by kind of framing the problem. We won't spend too much time here because we're all familiar with it, right? Problem is that drug development takes too long. From the time we identify an unmet medical need, we go through the discovery process and preclinical development and clinical development. That average time period is 10 to 15 years. And of course, the problem with that is that patient up there with that unmet medical need might not have that kind of time or have a poor quality of life throughout that period. On top of this, we're not really moving in the right direction. So timelines, cycle times for drug development have gotten longer. This data is a little bit old, but some CMR data up in the top left corner there shows average development times from 2001 to 2010 increase from about 12 and a half years to 15 years. So recently, I think that's leveled off a little bit. It certainly hasn't continued to increase at that rate. But we understand the problem, right? That's a long period of time. And we have all these new technologies and data and analytics and all that type of stuff. And we can apply that in a lot of different areas, right? I have a cell phone in my pocket with about 30 apps on it that help me do things better and faster. But drug development is complex. It's very complex. So we start looking at what are the different factors that are impacting the length of drug development, kind of naturally gravitate towards clinical as one of the big areas of focus, right? It's the longest, costs the most amount of money to put a drug through clinical trials. We have some data here from PHRMA that kind of sheds some light on the issue. The complexity of clinical trials has increased significantly. Right? Trials are more global in nature now than they were before. And there's some different metrics here in terms of the total procedures per trial protocol and trial treatment period, number of case report form pages per protocol. A lot of these different measures here all pointing in the same direction, that clinical trials are getting more complex to execute, and that's a big part of the reason why drug development timelines are as long as they are. But never fear, IT to the rescue, right? IT can solve this problem. Data analytics, we got it. I'm kidding, of course, but I think all of us in this room share the belief that IT and data and analytics can contribute to solving some of this problem. This graphic is from an article that McKinsey published 3 or 4 years ago talking about IT and the different ways that IT can contribute to speeding up clinical development times. And you see there's ways to leverage IT and data in all the different areas, from design and planning all the way through the report. And in different areas, clinical data management, document management, project and resource management, there are tools and processes that we can implement from the IT perspective to make progress in this area. But one of the things that I've seen missing in this article, a lot of articles I read, talking about the data and analytics and the benefits, is that most of these analyses don't take into consideration something that Bob mentioned in his opening remarks this morning, and that's the complexity of the R&D ecosystem. Right? So it's no longer the case that the typical process is big pharma company discovers a drug, develops a drug, and commercializes it. Now it's much more of a collaborative process. We have government institutions, we have academic institutions that might be involved in the discovery work. We might have a small biotech company doing the early development work with some support from a venture capital firm, right? And then maybe there's a collaboration or a partnership with a big pharma company to do the clinical development work, and they outsource that work to a CRO, right? More than half of all work now, clinical development work, is outsourced to CROs, right? So the R&D ecosystem is more complex than it's been before. And my background, and what I'm going to focus on, I have a talk later today, is that interaction in clinical development between the sponsors, the companies conducting the work, or sponsoring the work, and the CROs and the providers that are supporting development of that work. Because that's a very important part. As I said, more than half of all clinical development work is outsourced now. So we have these two ideas, the one being there's data and analytics that are giving us some new useful, powerful tools that we can use to enhance drug development, and the idea that more of the actual development work is being outsourced. So these two things need to work together for us to make advances here. One of the things that we looked at in the research that the Evoca Group, my company, conducted earlier this year is, how is this working right now? That relationship between providers and sponsors, how is it working in terms of leveraging what we called intelligent clinical development approaches, right? We had a definition for that in the research that we conducted. We did some research, we talked to a bunch of sponsors and a bunch of providers to get their opinions on how well it's working. So you see the results of one of the questions that we asked, which was, to what extent does your company's clinical service providers either contribute to or detract from your company's use of intelligent clinical development approaches in each of the following areas? And you see them listed there: site selection, region selection, monitoring, patient recruitment, all these different areas. And it seems like in some respects, in some areas, we're making progress, right? Site selection, region selection, you had a majority of sponsors indicating that their providers are helping them make advancements in this area. But for 6 out of the 10, the bottom 6, starting at project management all the way down to cost benchmarking and management, what we had was a majority of sponsors indicating that their providers are making little to no contribution in these areas in terms of the application of intelligent, data-driven approaches, right? And even in some respects, a negative contribution. So there's a big opportunity for improvement here. Now, I tried to find a couple success stories to kind of reinforce the potential, right? If these things come together, if the idea of data and analytics We can leverage that to streamline clinical development, and more that's outsourced, so we need to take into consideration the partners that are going to be contributing to this. I tried to find some case studies, and I got to be honest with you, it was hard. I found two, one from Covance, one from Quintiles, and full disclosure, I used to work for Covance. They had the best example I could find. They have an Accelerate platform, which is basically their analytics platform they've built over years and years that helped them conduct clinical trials. And according to the case study from Covance, they say they were able to leverage this Accelerate platform to deliver a pivotal oncology study 1 year ahead of schedule. Powerful stuff. Quintiles has their own tool. It's called Infosario. Again, they've been developing that for years. They say, partly due to Infosario, they've been able, over the last 3 years, to deliver for 87% on-time enrollment rates for their trials. And last I checked, across the industry, more than 50% of trials failed to meet their enrollment timelines. Right? So the potential for big advancements here. But these are just a few examples, and I think there could be many more over the coming years. So as a quick summary, if the goal is to accelerate development of new therapies to the patients who need them, then from my perspective, one of the key areas that we need to focus on is the design and implementation of systems and approaches that help sponsors and providers collaborate more effectively to leverage data and analytics to streamline clinical development. And I'll turn it over to Michael, I believe is next.
Thanks. So I'm Michael Cantor. I'm from NYU. I used to work at Pfizer before that. And I'm going to go over to probably the other end of the spectrum from the last talk, and I talk about the patient perspective and actually how it works, how these kind of ideas around personalized or precision medicine can actually work when you implement them in the clinic and what we actually need to do that. So the first issue is around working with data. And having worked in industry where we really work with clinical trial data, moving over to a medical center where we work with electronic health record data, the data sources are really difficult to deal with. You know, a lot of these are built up, particularly at Pfizer, from legacy companies. There's not really a lot of standards built behind them, and if there are standards, they're kind of our own individual standards. So making them analyzable on the large scale is a really big challenge. And then once you make them analyzable, how do you actually, so you can actually understand what the data actually say, so that if I have an F and you have a female and you have a 1, we all know that those actually mean a female patient. How do you actually put them together in a meaningful way? One of the ways that we're trying to do that, one of the projects that we're working on at NYU, is a project around personalized prevention. This is Scott Braithwaite in our department basically saw that when you go and see a doctor, one of the interactions that you have, particularly in primary care, is around your preventive care. So should you get a mammography? Should you get a colonoscopy? Should you get a vaccine? And the U.S. Preventive Services Task Force has about 40 recommendations that are rated A or B, that are actually paid for in the Affordable Care Act, and so that are applicable to most of the adult population. So how do you decide what to recommend among those 40? So the project that Scott worked on is actually to figure out, using mathematical modeling, which of those interventions will give you the highest, the best outcomes in terms of life expectancy. And so that's one of the ways that you can prioritize, so that you're not going over 40 different items at every visit. We're working on implementing that, so that we've implemented that in a pilot. We're actually now working on implementing that on a mobile platform and actually trying to do it in English and Spanish so that's applicable to patients across the spectrum. And this actually brings me to that last bullet there to kind of frame this in the perspective of a patient. So when I worked at Pfizer in 2008, I had a panel of patients. I worked in the Bellevue Medicine Clinic, and I kept up working there throughout my time at Pfizer. But I moved over from having my own patients to the walk-in clinic, and I had 3 patients that actually stayed with me throughout, that I would see them every probably 3 to 6 months. And one of them was a 58-year-old lady. Her daughter was actually the same age as my daughter, so they both went to public schools in New York. We would always talk about it, but she didn't speak English. She didn't have insurance. She was actually here without papers. But she had a couple of medical problems that were not obviously too acute. They were generally chronic. She had hypertension, hyperlipidemia. But she also had this persistent anemia. Her anemia, her hematocrit level was in the mid-30s, which is a little bit low for, actually, the low 30s, a little bit low for a female her age. And so she had a colonoscopy, didn't find anything, did some other studies, really didn't find anything. Luckily, she wasn't symptomatic from it. But the question is, so what do we do about this? How do you actually, do we just kind of keep monitoring it? Do we go and do some genetic testing to figure out what's going on? And how do you do that in a resource-constrained environment? And how do you use all the data that we have in our systems to kind of, do the best care for this patient? So one of the things that would be helpful for this is around clinical decision support. And this is some of the work that we did when I was at NYU before I went to Pfizer, was around trying to build a platform for clinical decision support that would be platform independent. So clinical decision support is very basically, so if you're in a doctor's office and the doctor is there with his electronic health record, or it can be with any system, but they're telling you, giving you alerts or giving you advice, or here's a patient with X, Y, and Z. This is the kind of treatment you should give, or this patient's over 50, has never had a colonoscopy, you should give them a colonoscopy. Kind of typical functionality of an EHR system. What we were trying to do, and what ARC has done, but funded through the government also, is try to create a platform where you can have this independent, almost like an independent server that's platform independent from the other EMR systems that would get patient data and send back alerts or send back recommendations. And it wouldn't matter if you were on Epic or on eClinicalWorks or whatever. They would just get the data and send it back, and then you can interpret it your own way. So ARC has put a lot of money behind this, but because of the way our EMR system works, with there's so many different vendors, so many different systems, hasn't really made it out to the mainstream yet. But the question is, how do you incorporate all this into a 15-minute visit? And particularly if I'm dealing with my 58-year-old woman, do I talk about her colonoscopy? Do I do my personalized prevention, talk about her vaccines, talk about her headache, about her, her daughter's problems in school? Or do I talk about, her anemia, again, which is not bothering her, but it's kind of bothering me because I see this number and I don't really know what to do with it. And so when you're developing these more sophisticated clinical decision support tools, and when you get to personalized medicine, ideally you'll have data not only from the typical genetic data that's out there right now, which is generally from Northern European populations, but you have information from populations that are very similar to the patients that you're seeing. You can also have epidemiologic information from patients who are similar to your patients as well. I think combining those two is really where you get the benefits of that. And then you're able to prioritize not only around prevention, but actually on your clinical interventions for patients too. And then you make that patient focused, and that's basically what I'm talking about, is tailoring this for particular patient populations. So again, it's very different from the patients I see now. So since I moved over to NYU, I'm in private practice. Everybody has insurance. Everybody speaks English. And I've already had the stereotypical patient who's coming in with their hand printouts from the internet about the 7 different problems they want to talk about, and are sure that they've got all the right answers already. So as opposed to my other patient who I worked with at Bellevue, who just kind of wanted to chat and kind of was really ready for the very probably 1950s version of a doctor where you're a lot more paternalistic. And so it's been a very interesting cultural change for me also. But some of these things, when we think about personalized medicine, oncology is really where it's been booming, and particularly in the pharma industry. Pfizer's Xalkori, for example, that's really where a lot of the benefits have been, and we haven't really moved it out of oncology yet. I think that's kind of our big challenges there also. But also in making it patient-focused, the way to get people to adopt things in medicine these days is generally to link it to a quality measure. And so, if you think about how a lot of behavior has changed, particularly in primary care, it's around these quality measures. So you're measured on how your diabetics are doing or how many colonoscopies you're giving, and incentivizes the doctors and the healthcare systems to make it easier for people to get this kind of care. The question, though, is that how do those quality measures correlate with patient satisfaction? Because if you want to be patient-centered, you have to think about that in the actual clinic visit, about what the patient is wanting. Like Bonnie was saying, it's all about them. So, again, the patient who's coming with her printouts from the internet versus the patient who, just wants me to tell her what to do are very different. And what their perception of satisfaction is going to be very different as well. And the last thing is trying to get to these patients with chronic diseases. How do we think about doing genetic testing? Are there kind of more, potentially more expensive, potentially more complex, potentially exams that will open up different types of information that maybe you don't want to know, and in patients who are uninsured and patients who have different priorities. So I think that's really the biggest challenge in adopting personalized medicine or precision medicine, is figuring out how all this relates to the patient priorities. How do we adapt them for different types of patient populations, and actually how do we link that to quality as well, so there's good incentives for them to be adopted as well. Thanks.
Okay, my name is Mariano Alvarez. I'm from Columbia University, and I'm going to show an alternative view and approach that we are applying at Columbia University for personalized medicine. So the current view is to look for what is broken, especially in cancer, if there are mutations or DNA amplification that can be used to decide which genes or proteins to target, and go for them. The problem is that if we take a population of patients that they show a very similar phenotype, for example, luminal breast carcinoma, we will see that there are a huge variety of modifications and genetic alterations that leads to a very similar phenotype. So what we propose is that there should be some mechanism, regulatory mechanism in the cell that integrates all of this kind of information into a given defined phenotype. So we propose that there's an integrative machinery that is a regulatory machinery in the cell that integrates changes in the genome, but also integrates information from the environment to direct a change in the phenotype that induce tumor progression or metastasis. And for approaching this, what we do in the lab is reverse engineer regulatory networks to try to identify which are the set of regulatory proteins that integrate this information and make a similar phenotype from a different variety of alterations. So I will give an example of which is the approach, and I will show some real data on how we can use this on personalized medicine. So our approach is reverse engineer regulatory network that contains all the transcription factor, all the genes that are known as transcription factor in the cell, and the target genes, and use these to infer which are the proteins whose activity is modulated by changes in the phenotype that are associated with the progression of the disease. And in parallel, focusing on the therapy, we can generate gene expression profiles from drug perturbation assays and identify which are the proteins whose activity is affected by these compounds. And by measuring the similarity between the effect of the compounds on the regulators and which are the regulators that are involved in the disease, we can finally find single compounds or combination of compounds that can block the activity of the proteins that are upregulated in the patient and induce the activity of proteins that whose function was lost in the patient. So as an example, I will show some data that we generate at Columbia for a particular patient from which we have data from the primary tumor and several metastases in the liver. The tumor is a neuroendocrine tumor of the colorectal tract. And what we did was generate genomic data, including genetic and transcriptome data. And we collect in the course of the last 2 years 300 fresh frozen samples from neuroendocrine tumors. We profile them and we use the algorithm that we developed in the lab to reverse engineer the regulatory network. And by using this network and the samples from the patient, we identify which are the putative tumor dependencies, which are the regulators that are involved in the transition from primary to metastasis. And in parallel, we are generating drug perturbation data from cell lines that are neuroendocrine, that were originated from neuroendocrine tumors to identify which are the putative compounds that we can use to block the activity of these regulators, and after the in vitro validation, move to a preclinical study and finally to the clinic. So this shows the cluster analysis based on expression data for all the 300 samples that we collect and profile. And what I want to point out here is that we have very discrete clusters some that are enriched in neuroendocrine tumor from the pancreas, some from the small intestine, and some from the rectum. But one important thing is that in this cluster, we have together primary tumors with the metastasis in the liver. So we know that the phenotype of the cell still maintain the phenotype that was original on the primary tumor. So we use our master regulator analysis algorithm that I'm going to show and explain in a talk this afternoon to identify which are the proteins whose activity is differentially active between the metastasis and the primary tumors. And we compare these results with our inference for the regulators from different cell lines to select which is the cell line that best recapitulates regulatory mechanism in the patient. So we know which is the best model to use in the in vitro screening assays for look for compounds. So this shows some results of selected compounds, and we select these ones according to their effect on the regulatory machinery of the cell model that we are using. And this shows the minus log of the p-value for the effect of the compound for inhibiting the regulators that we identify in the patient. So we can see that we have a few compounds, compound 1, 2, and 3, that has some effect on inhibiting the regulators that we identify from the patient data, while there are some compounds that has no effect. And when we evaluate the effect of these compounds on xenograft models using the cell line model, we see that the compound 2, that was one that showed the strongest effect as inhibiting the regulators that we identify specific for this patient, show the strongest effect inhibiting the growth of these tumors. So, thank you very much. And I pass this to Robert.
It was on the drive. You copied it to your hard drive. No. Bonnie, play some music for a second.
You can put on the Jetsons.
There you go. Put on the Jetsons.
You want to put on the Jetsons? Nope. They're slow at the draw.
Field a question or two?
Anybody have a question or two they want to quickly interject? No burning questions? Okay. Before we get to mine.
Okay. All right, thanks, guys. Sorry about that. I thought you had everything. So hello again for people who were not part of the morning session. We talked a little bit about some of the things we're trying to do around bringing the data in and facilitating the research to actually move the data into a position where it can become more part of what we're talking around personalized medicine. And if I had the opportunity, I probably would want to grab each of you after this and say we should be joining together on this kind of platform, but we'll take that later. So, this was a presentation that was actually presented back at the Jamboree in New York at the Mount Sinai School of Medicine. And it's around JACE. And JACE, we talked about the platform of JACE, and we're talking about a deeper dive here around the JACE platform. And again, JACE stands for the Janssen Collaborative Environment. So again, the incarnation of the JACE platform came by way of and I talked about collaboration this morning. We're big on collaboration as an external innovation group, and we liaised and we partnered with a company based out of Atlanta, Georgia, known as Liaison Healthcare Informatics. And it was a rapidly developed system. It's a very successful system in our minds, and it's a growing system. The underpinning of the JACE environment and the goals of the JACE environment are that we want to use Transmart, a translational database, coupled up with a index, which is a national data exchange, or not, I'm sorry, the data exchange platform that's actually part of the JACE platform. and I'll get into that in a second, but the JACE platform itself is interesting. We looked at our partner, Liaison Healthcare Informatics. It's just not a hosting company. We wanted to make sure that we're building something into the platform as far as a plug-and-play functionality would go. It's one thing to put data into the JACE platform, but it's a whole other thing that if 5 partners came in and they have 5 separate data sources that we all wanted to share, we also might bring into the system 5 sets of tools that we want to use. So we have this plug-and-play mentality. You bring your data and you bring your tools, and we all share the wealth, and we get to utilize these tools and share the data. So this particular schematic just basically is an architecture of the actual system. So you have a monitoring dashboard, you have the JACE landing page, obviously from a web front end, but the guts of the system really revolve around this Transmart index and what I call future applications, whereby we are able to plug and play with these applications and drive the analysis of the data to even a deeper level or a further level. And obviously, there's some file sharing capabilities, and there's the operational visibility and dashboards. So what does this all really mean? So if you look at the bottom of these data sources, we're going to really kind of think about these things. Continuous unstructured data. So it's a feed like a social media feed. So we have RDM, we have other file formats, we have image data, and we have other types of data. So the goal for JACE is to be ubiquitous. The goal for JACE is not to be just a one-off, one-stop shop for people to kind of get in and do their research. It's an ever-growing, evolving ecosystem of data sharing and tool sharing among the partner companies. And the data is very important. Probably the most important thing about the data is that it doesn't have to, I think one gentleman had a question earlier today, or maybe it was Mike, around the problem of, yeah, this is all good on the surface. You can get through the politics of sharing data, but when you get into the data, the guts of the data is disparate, it's different. How do you work with different types of data, and especially around, I think Mike had a good point around electronic health records data. So maybe that'll be something that JACE can tackle in the future, but today it's really just getting started with JACE. There's a couple phases to the JACE platform. So phase 1 was really around getting into the position where we can have external links to the solution. Desktop virtualization, Transmart, and Index are the underpinnings of the JACE system right now. And then hanging off there, you have the NSF, the FTP, and the web browser. Data harmonization is a huge part of what we're looking for out of our partner, Liaison Healthcare Informatics, because we can't do this alone, and we know that they have the ability to move us forward in the data harmonization challenge. And we talk about third-party data, application database data. Mike had this point, EMR data, PHR data, social media feeds, and big data, which is a big topic of the conference. So if you put all this together in a nice, neat package, it's a way for us to engage academic partners and industry partners to say, bring in your data, bring in your tools, and let's analyze this together in a collaboration. So Transmart, which is part of the JACE system, I was hoping to take a little bit of a tour with us earlier today, but we only had so much time. But Transmart, again, for people who weren't part of the meeting this morning, it was a system that was created by J&J. It was evolving and maturing, and then obviously it went into an open source posture for the company, and we moved it out into the open source world. And it's been successful. There is a Transmart Foundation that is built around this platform, and I'm sorry, you guys are kind of craning your necks, and the monitor's not high enough, so we can share these slides later, or you can all turn your chairs around maybe. But suffice it to say, the idea of it was to release it in a version 1.0 and then have the developer community attack it, not hack it, but attack it for building it up and making it more mature as the source code goes, as the source code base goes. Again, this was from this morning, but another just quick overview around Transmart. So the resource is provided for or by the Translational Informatics group. So you have a group of computational biologists that are leveraging the JACE platform now, and the Transmart platform that's the Transmart application is part of that. And really actually is just developed by us, I mentioned that already, but the data repository is growing. So we have a subject level clinical trial biomarker data for internal, external public data, approximately 150. By the time I printed this, it's probably up a little bit higher since the last presentation on this, and around 4,500 for the results of level information for clinical, non-clinical gene expression analysis, and there's curated literature as part of it. So computational biologists can not only see the data, they can get in, they can actually work on hypotheses, and they can actually work on they can see papers that other researchers have published that are also part of the actual platform, part of the Transmart platform. I'm trying to go quick here, guys. I know we got 8 minutes, so. So what's the rationale around the Transmart story? So it's really around the rationalization of what can we do around a couple things. what are the options that we had back then and what we're considering around this platform. So they were to take either an extreme position of do nothing and kill the project and just think it was not going to be anything more than what it was for a one-off internal project, or we were taking the other extreme, kill the project, replace it with a commercial offering. We didn't do that. It's open source. Joint venture with a software company, we didn't really do that per se. Spin it out as a company, it's really not that. It's open source. This is the key. We released it under an open source license, and being a open innovation, pre-competitive mindset, philosophy, this is the point that we're at right now. So does it have real value to others? Well, yes, it really does. So our industry partners are actually using this, and the idea here is that for us to be successful, we need to research and talk to a partner. Somebody mentioned earlier today, how do you bring a new partner on board? How do you make sure they have the knowledge and the ability and the skill to be part of the system? Well, you do your research and you do your vetting of this, but what you want to do is make sure that they have a similar problem. It doesn't make sense for them to come into Jace and into Transmart if they don't have the same problem that we have. So at this point in time, early experiments were run around the academic consortia, and we've actually partnered with IMI, a project in the Imperial College in London, and St. Jude's Hospital, Research Hospital, and that J&J relationship is very successful. J&J provided a license to the consortia to use the software basically for free. So a quick overview, the motivation to undertake this for translational informatics. So Transmart really is around data management, knowledge management, and translational informatics. And the idea here is to improve the drug R&D productivity. We talked a little bit earlier today around reducing cycle times, increasing efficiencies around this. So really, how do we improve that? Well, a couple ways. We look towards efficiency, and we look towards effectiveness. And there are the two key tenets of actually running in this system. It doesn't make sense to create a system that would bring in all your organization's data and just have it be a behemoth, a very lethargic process. We're trying to really make it speedy and efficient. Other challenges around running into the internal biomarker program were these, study populations defined by a clinical development program didn't really provide cross-sectional view of data, right? And we really didn't have a early detection system built. So we wanted to put this into and make this part of the DNA of the Transmart system. So the cost of running sufficiently powered phase 0 studies is prohibitive. We knew that. And also to overcome these challenges, we were focused on the two key trends we're chasing in this conference: big science around big data collaborations and consortia, which I talked about this morning, and about next generation technology for characterizing biology. So all this said, Jace coupled with Transmart coupled with Index is the direction we were trying to take. And Transmart really is the bread and butter of the system right now. So how do we really organize with collaborators and do this across institutions? We face that challenge. We think we're pretty good at that by now. And how do we make the data available to all people and all the partners? And really, it's around this type of structure. It's pulling in clinical trial data, non-clinical gene expression, curated content, text corpuses into this data warehouse that we saw this morning, and then making master data, federated data part of it, and then having key, strong mining tools that actually are part of the platform known as Transmart. This is what Transmart looks like to the person who would actually use it. I won't go through these screenshots too deep, but it's very easy to use. There's a drag and drop functionality. It's very effective for the computational biologists to use. They can do quick queries on the data, and they can spit out quick renderings via Spotfire. So Spotfire is part of it. And they're actually getting to the root of the data analysis in these screens. It's easy to create a hierarchical model with the ontology system that's part of Transmart, and that's an easy to use. It's a fast, effective way to kind of get data in and use it. It has a faceted browser capability for people who are out there that use faceted search. It's pretty strong. It's robust. It's been proven. And the conceptual architecture, again, is pretty simple. It's just basically Transmart web app sitting on all the results database, clinical data model, omic data model, gene pattern, R&P link. And it wouldn't be anything if it didn't have curated data. If Transmart did not have curation, it would not really be up and running and successful. So the aim of Transmart really was to build a high-quality, reusable resource around curation, make it accessible to all users, and make it open source. So the experience now here is that they really understand how to implement standards within the platform. We can spot errors quicker, and we can organize data faster for reuse. And data owners are often not so good at this, but Transmart makes it a little bit easier for them to get better at this. This is the story of the orientation, the story of the way to get data into Transmart. You would define an ontology. You would convert the data of the ontology into formatted specifications. You would transform that text and then copy files to the I2, B2, and DF tables. Long way of saying that it's a path forward to get data into the database, but once it's sort of done a couple times, it's not too hard to do this. Again, just a screenshot around the actual graphical user interface and how easy it is to use. You would map the files to the data file. Map the data to files, and then the running and loading of scripts. If anybody's interested, we can go into a little bit more. I know I'm going faster because we only had about 8 minutes apiece. I think the main key thing here around the ease of use of the Transmart platform is really not so much around, asking people to join us and be part of an organization and part of a collaboration, but really it's putting the problem first, putting Transmart back out and saying, what is your problem? Do you have a like problem? Well, here's a tool that you could leverage and use for the greater good. So really in closing, I won't belabor this. We already went over this slide, but really it's around this. It's around collaborative sharing, which I spoke about earlier today. It's around collaborative refining of data, and it's around collaboration, organizing, and measuring this data with the consortium members that are part of the foundation. And if you want to go to transmartfoundation.org, you can see more information there. And I think what I'll try to do in the spirit of time is move. We saw this today. And just talk about the benefits of the community, of the Transmart Foundation community. So you're actually seeing, just as a small snippet of the actual community, but it's always an evolving platform. The open source community is constantly building on the code base. And so far, I think in June 24th, I believe, or June 20th, there will be a second version of Transmart released to the public. And thank you very much.
So I want to thank our 4 panelists and take questions. Is there a mic?
So I have a question about Transmart. How do you go about prioritizing what goes into a release? So for example, if we were to try to adopt Transmart and there's some important functionality that we needed for our business to move forward, how would we get that into Transmart?
That's a good question. There are today roughly, I believe, 24 to 30 developers in the open source community for Transmart. They meet, they have summits, they listen to the businesses and see what the problems they face within the Transmart community, and they would probably look at that point where they would listen to you do a voice of the researcher, VOR, and then take your problem and see if it's also proliferating throughout other companies that are using Transmart, and then go from there. I don't think you can email the Transmart community and say, I have this problem, and you put this piece of code in for me. I think it's a little bit more than that. And I think it's in line with any other software engineering effort that you would do. You would listen to what the business has to say. You would sort of stratify all the pain points around the organization. But I think it really starts with the Transmart Foundation.
And can anybody get into this Transmart development community?
Yes, it's an open source community. If you think about it, Transmart is much like Linux. When Linux started with the I think the advent of the online, the open source community. So yes, it's possible. Yeah.
So is the business model is open? Does
Yes.
open mean free?
Yes.
I'm going to raise an issue which I consider to be the elephant in the room issue. We have a lot of basic science discussions. I'm trained as a basic scientist. I spend most of my time working with clinicians. I respect and depend on the technologies and the science that a lot of you have discussed. But the issue is, if you're be patient-centric, you have to understand how healthcare is delivered and what are the issues in healthcare. Unmet need is not the fact alone that there isn't a specific treatment. Unmet need may be a treatment that already exists, but the dosing isn't one that's acceptable to the patient. It may be a guideline that a physician won't use because it's too complicated for the patient. We talk about the disease issue, And one of the biggest problems in disease is diagnosis. It's staging a disease. The information that the physician has that was touched upon has to deal with the fact that almost every patient that shows up in the clinic, as was pointed out through a couple of anecdotal examples, has more than one disease. Because they have more than one disease or comorbidities, some of them are being managed with other medications. Some of them are not being managed or may not even be known. And all of these things are going to complicate anything that we try to do to intercede with the disease we're trying to tackle. We have to understand that the patient represents a much higher degree of complexity and that the healthcare system is much more complicated than we would like it to be, so that when we develop a drug, it automatically goes into the market, because if we make the investment to develop drugs, as we know is very costly, and we go through the process of having a drug approved, if a physician doesn't prescribe it and a patient doesn't take it, it's a success, but it's a commercial failure. And commercial failure isn't what pharma is about. And pharma has to understand all of these levels of complexity early in the process and not after the drugs are being developed. And I think these start to bring in, along with other things, issues of what a patient really understands about disease, because what's acceptable in an oncology situation is very different from what's acceptable in a cardiovascular situation, a GI situation, a CNS disorder, or psoriasis. And all of these impact the entire, that should be incorporated in our early stage thinking about drug development, because it will impact not only everything through the development and trials, but actually whether these drugs go out on the market and produce benefit to the patient and success to the company.
So I can begin to address some of those questions. I would encourage all of you in this room to go on the patient communities, and you will be surprised at the level of information and knowledge, especially smart patients, which has micro experts, is how it's described. And in answer to the question, patients are driving many of these research decisions already, and that's kind of part of it. I think another part of your question has to do on a data side with a question I had for Michael in terms of clinical decision support. If we're going to have these more complex algorithms, machine learning, natural language processing, that's going to help us to understand more of what we need at a personal level, how is that going to catch up, Michael, with the clinical decision support algorithms that were developed how many years ago?
Yeah, well, so I'll answer that first, and I'll go back to the other question. I mean, the clinical decision support algorithms are just algorithms. You're right, the EMR systems are based generally, a lot of them on very old code, but it's relatively straightforward to implement them. The question is, how do you actually use them in practice? Because, you know, if you have a patient with 7 diseases or, you know, 3 multiple comorbidities, you want to get 20 alerts about all the different things. So you have prioritization is a big thing. But I would say second, you know, the way you frame the pharma industry is kind of, you know, the outs. I mean, I can understand why you would say that, and there's our history kind of goes to that. But if you look at the areas that pharma is focusing on now, I'm thinking from the Pfizer example, it's cancer, neuroscience, CV and metabolic, and autoimmune diseases. And if you think about how those relate to comorbidities, if you have cancer, you don't care about your diabetes or whatever because you want to cure your cancer. If you have Parkinson's disease, that's probably your principal reason. The same thing as Bonnie was talking about. If you have rheumatoid arthritis, CV Med is a little bit different because it's mainly about the prevalence there and the need to get better treatments for diabetes or for hypercholesterolemia or whatever you're going to look at. So the way that pharma is developing drugs, first of all, they're responding to the market because their efficacy trials get you approved, and that's the goal for these drugs. But you're also thinking about effectiveness, and the strategic thinking has developed way more than it was 20 years ago. I mean, people obviously are doing a lot of epidemiological work and a lot of work before they invest billions of dollars into a drug development program. So the perception that pharma is just kind of putting out these drugs just to get them approved and then they might be a commercial failure may be relevant in the minority of cases, but I think in the vast majority they use competitive intelligence, we use epi, we use a lot of different mechanisms to make sure that it's going to be successful. And you think about your patient population and their priorities. So again, that's why you're focusing on these diseases that have really bad outcomes and really affect patients' lives, because I'll deal with my diabetes getting worse if it means that it's going to cure my cancer or get rid of my tremor for Parkinson's.
Or maybe at the opposite end of the spectrum, we're going to see more pharma companies looking somewhat like health and wellness companies in the prevention zone.
I would say that pharma thought about that. We certainly had a health unit. Again, this is my myopic view from Pfizer, from our integrated, and this is my opinion. This is not, I don't work there anymore. But we had this integrated health business unit that was going to go towards wellness, and there's certainly a lot of recommendations that people should do that. But the margins on on a wellness effort versus the margins on Humira, are just not, don't really fund your business. So I agree that we need to focus on that and take those considerations into how you formulate your drug programs. I agree that wellness is a huge issue. And ideally, if wellness was more prevalent, it would put pharma out of, and doctors out of business, because we wouldn't have a lot of these diseases. But I think it's very hard to make a business case for that from the pharma side.
More questions? Yes.
I actually have two questions, one for Michael since he just talked. I've been recently listening to a seminar from David Goldstein from Duke University. It was a fantastic seminar about precision medicine. And now listening to your talk, I was wondering when are we going to get sequencing in a private practice and what's going to be the impact on that?
Yeah, that's a huge issue, and I think that as the sequencing gets cheaper, that it'll be available. But then the question is, what do you do with that? What do you do with variants of unknown significance, that come up, and what's the liability, for an internist and who's going to try to interpret genetic, markers? You're not going to look at the sequence. You'll look at some markers and try to determine risk, kind of like a 23andMe report. I think is probably where you'll get to. But I don't, in my anecdotal view, and working in primary care, I think we're really far away from that, because the incentives aren't necessarily there, because we haven't really proven that sequencing a patient, looking at their genetic markers, will change your treatment to a point that it's clinically relevant in most areas. In cancer, obviously that's kind of standard of care now. But if you think about for diabetes, hypertension, the chronic disease, lower back pain, there really aren't the tests haven't shown the impact out there.
He brought two very compelling examples of disease that sick patients that they didn't even know what they had,
right.
and they came up with
right.
with the therapy in the span of 6 months.
Yeah, those are anecdotal. Those are
rare.
Right.
These
I know.
are
Exactly. Like there's the kid in Wisconsin who had like 1 million surgeries, and they finally sequenced him and found out this. I forget what it was, but this enzyme deficiency or something. But you're absolutely right. In the very on the extremes, it'll be there. But and again, being in 90% of my patients are chronic diseases that I don't need to sequence them for. A lot of it is behavioral modification. So. But yeah, I think there's always going to be those cases where it gets a lot of press and it's amazing, but it's a small part. Luckily for us, it's a small part of the population. But again, I think in cancer standard of care, I think it's going to take a while to disseminate, because you have to prove the value on a larger population. On the rare cases, the NIH has an institute for a program for undiagnosed diseases where they fly people into Bethesda and sequence them and try to find out what's wrong with them. So that's certainly a tool, but it's still a niche area, in my opinion.
And then I have a question for Robert. You brought additional examples of pharma outsourcing stuff. We already knew that pharma are already outsourcing research by buying companies or acquiring their pipelines. How do you envision big pharma in 10 years, if they're just going to be aggregators or what?
Good question. Okay, so personally, for me, the way I would look at it would be, and this is the George Jetson analogy, or where Judy would be. Me, I would think from an innovation standpoint, from an IT standpoint, that we are working together. And what I mean by that is that you're never going to have a situation where you don't have large monolithic institutions and big pharma companies. But I think what you're going to see is a more of a harmonization, if you will, around this landscape, this ecosystem that we're talking about and working together. 10 years is a long, actually it's not a long time, but 10 years isn't such a long time. But in the grand scheme of things, I think you'll see more partnering going on. And it's a trend. I'm not telling any secrets hereby. We know that we're partnering more and more these days, and we're reaching out to academics and other institutions and also industry partners to be part of what we do. Part of what I showed earlier today around spreading the risk, rising tide floats all boats sort of philosophy. The speed, I think, is the key at which that happens. I think the acceleration point of where we are today and where we will be in 10 years is going to be a true key of, does all these collaborations, do all these accelerated paces that we can put, you can throw all you want of the technology that you want in a problem. Michael had a good point around, yeah, you can have all this technology, but really what's it about? It's about the patient. So it's moving, I think, the technology in a few different ways. You move technology into the bench, help the people there, but you also move technology into, and we see on-body sensors, we see Fitbits, and we see Vivo Fitbits and things like that, and people wearing patches in these Garmin the advanced Metria patch on the back of an arm, taking biometrics from a person. I think you'll see more of that. I think that would be part of it. I think we're doing a study now around the feasibility of using on-body devices and what's the obtrusiveness versus the unobtrusiveness in that model, and do people rip them off and say, I really don't want to wear this? It's really it's a uncomfortable thing to be wearing, or will they actually embrace it? So I think you see that trend from taking wearables and fitness and also collaboration and maybe blending a little bit of that together. It sounds like I'm talking crazy, but you're taking an inflow of data into this situation and also collaborating with these other companies that are part of that equation and using that for personalized medicine. And also I think you would see much more collaboration than we do today. Yeah. Sure.
So on your point about wearables, I think what you got to do almost is talk to the people who make phones and Google Glass and stuff like that, and figure out how you can get some of the technologies that you need in those devices, because that's how you're going to get. People only want to have a certain number of things around. Like, you want to. Do you guys have plans around looking at how you could leverage Google Glass or the stuff in a phone to be able to not require people to have excessive wearables or expensive wearables?
It's interesting that you brought this up. I'll take the Google Glass example first. I mentioned earlier today that part of my job early on was to actually sit and do cycle time efficiencies with scientists and see what they're doing in the laboratory, and sit there with a stopwatch and see how long it takes one cycle from one spot to the next, one station to the next, and see if IT could bring anything to bear on that challenge and create an experiment around that, around technology. One thing that we're working with is that typically there's a recipe in a lab, and a person will be looking at a hard copy printout of something, and as they're moving, they're using their hands, they're picking up the paper, they're opening the book and changing that recipe page. Well, why not have the glass scroll? I think we talked about this earlier today. Why not have the glass scroll the recipe in front of the person so that they're basically not having to touch anything? It's just right there. They can speak to it. They can speak their findings. They can actually interact with the Google Glass. We're actually running a proof of concept around that now. As far as wearing something goes, I mean, a Vivo Fit is not heavy. It takes a lot of step counts and things like that, but I don't see that where it's really that obtrusive. There is a company out there called, I believe it's MI2 or M2, if you look it up. They've created a device that looks like a Band-Aid, but it's got a computer chip on it, and you put it on the part of the body that you're trying to get a sense off of, and they're extremely low profile, they're extremely lightweight, and they're using them for babies. They're putting them on a chest of a baby now to detect heart conditions, and I thought that's a cool advancement. Maybe the Fitbit one day would be just a Band-Aid on the arm. Nothing more, nothing less, nothing that obtrusive. Maybe, probably, in George Jetson's time.
Did you see the ad on television for Apple's Health Kit? No, I haven't. Well, it's out there, and they're wearing wearables.
Of course, I use DVR and I don't see any ads.
That's a good question.
That we'll wrap it up. We have talks at 1. I will note that we are in the age where I was just at the doctor's yesterday. They're still sharing paper on one hand, and now we have people talking about using Google Glass to see data on the other. So this is a very interesting time where we're seeing a mix of old and new. With that, let's adjourn, and we'll come back at 1 o'clock for the different talks in different rooms. Thank you.